Good morning, micro classes. This is Professor Foss. We're going to be going over the Exam 2 study guide. If you have any questions, specific questions after I go through this, please feel free to email me or Professor Dirks, and we will get back to you. I'm going to go over this. Some of it is pretty self-explanatory, or I can just kind of point you in the general direction of the answers. And then some of it, because we didn't get to lecture very much, we haven't this entire semester been able to lecture very much. I might go into a little more detail. So let's get started. So one of our most recent labs, number 1, we were talking about if you look at blood agar for alpha, beta, and gamma hemolysis. So remember that hemolysis, heme means having to do with the blood, and lysis is to break open. So when we talk about hemolysis, we're using these blood agar plates. They have sheep's blood in them, and certain bacteria will actually break open those blood cells, and then you can look at it and see, is there a halo around it? If there's beta hemolysis, you're going to see a clear halo. It's complete lysis of the cells. If you see alpha hemolysis, it's just going to be partial, and it's going to look like a greenish gray or a green yellow. And if you see gamma hemolysis, there won't be any halo around it at all. So those are the ways that you can look at a blood agar plate and see what type of hemolysis has occurred. Remember that we were using this when we were discussing strep. I believe that's lab 8. Okay, so number 2, what do we use a McConkey agar to identify? This is part of our most recent lab, just yesterday. So remember that McConkey agar is, we're looking at a bacteria's ability to ferment lactose. There were 2 different results. If you remember the chart that we drew on the board, you could see E. coli on the McConkey agar. It was like a bright pink because it's an acid producer, whereas everything else, all the other gram negatives, will come across as a light brown or a tan, okay? So make sure that you review the charts that we made yesterday because they are definitely testable material. What are the different sterilization techniques, and know when to use these techniques? So we've been talking about this all semester, because we've been kind of trying to get you guys to understand what's the difference between sterilization and like other forms of aseptic techniques. So for example, sterilization, remember that this isn't something that you want to do on human skin cells. You don't want to sterilize a patient's hands because that would actually destroy their skin cells. So examples of this would be, autoclave or radioactive ionization or disinfectants. You could say like flaming your loop would be an example of sterilizing techniques. So know when to use these techniques. So like if we were talking about flaming your loop, that is a sterilization technique that we use when we are going to plate some cultures. Think about in a hospital setting, what would we use to wipe down a bed after a patient left? Just understand when to use these different sterilization techniques. What are physical agents? This can be found on page 57 of your lab manual. It's a bolded term, and that's something that you guys can just look up as you are studying for this. How can we differentiate between streptococcus and staphylococcus? Okay, so could you differentiate between these two using a symbol stain? Could you differentiate between them using a gram stain? basically you need to know, what are these? Are these gram negative? Are these gram positive? What do they look like? Can you tell what they look like based on if you're looking at a microscope slide? So, I want you to think about this in terms of if you're working in a lab in a hospital and somebody comes in and you don't know why they're sick, but you take a culture, like what lab techniques can you do to try to figure out if what you're looking at is streptococcus or staphylococcus? And then 6 kind of factors into that. What is the gram stain used for? What will the gram stain show us? Okay, and that's, we've been doing that for almost the entire semester. I think it was lab 3 that we did a gram stain. So, 7, does concentration influence how effective a disinfectant is? What about time? So remember we did a lab where we were observing petri dishes that were divided into quadrants, and there was like 0 minutes, 5 minutes, 10 minutes, 15 minutes. And then on other petri dishes, there were varying concentrations. So, what did you observe? Did you see that time was a factor, or concentration was a factor on whether or not it was killing the bacteria? What types of bacteria may be resistant to disinfectants? So there is one bacteria that we have talked about, or one type of bacteria that we've talked about a lot this semester because it's kind of just a plague on hospitals, and it causes MRSA, so that's your clue. But this is, MRSA is methicillin, excuse me, meh, but I can't speak right now. It's MRSA is something that's caused by a very resistant bacteria to antibiotics. So I don't want to give you the name because that might give it away, but looking in lab 8 would definitely help. So um, antibodies. We kind of talked about this in some of my classes. I don't know if all classes had time, but antibodies are basically Y-shaped proteins, and they attach at the Y-shaped side to anything that your body considers pathogenic. So remember that they can actually they can bind to 2 different things, one thing on each side of the Y, right? And remember that when that happens, it causes clumping called agglutination. So antibodies bind to things that the body considers pathogenic. Okay. Which species of Staphylococcus is the most pathogenic? What are some of the things that can cause? This is reflective of one of our previous questions, so I'm not going to go into great detail about it. But if you do look in Lab 8, you'll get these answers. So remember, you're not just answering what species of Staphylococcus is the most pathogenic. You also want to know, what symptoms can it cause? What can actually happen to the patient? Why do we use mannitol salt agar? Remember that mannitol salt agar is one of our selective medias that we just used in Lab 8. Remember that it will allow certain species like Staph aureus to grow, but it will inhibit other species. Okay, so question 11 kind of is lumped into this as well. If you need more information, it's on page 78. So, selective media inhibits the growth of some microorganisms while allowing the growth of others. Okay, so selective media, like mannitol salt agar, it will allow certain things to grow and it will stop other bacteria from growing. The differential media Differential media allows the growth of more than one type of organism, but results in observable morphological differences between the colonies. So remember, we just looked at our slides yesterday of our mannitol salt agar, and I know that a lot of you had kind of like a glowing Staph aureus, excuse me, and then people didn't have a glowing staph epidermis. Remember, the epidermis on your plates just kind of appeared dull, so it allowed both to grow, but morphologically you could look at them and tell that they're two different species. Okay. 12. This is also from lab 8. Catalase test. What enzyme is present if you have a positive result? So for catalase to show a positive result, remember you need the enzyme catalase, okay? And catalase is going to break H2O2, or hydrogen peroxide, down into water and oxygen. And remember, that creates bubbles. So remember when we did this in lab, we were looking at our slides, and you had to plate some bacteria and then add a little bit of catalase, or sorry, you added a little bit of hydrogen peroxide, and so if that bacteria had catalase, then you observed bubbles on your slide. Okay. Yeah, so what does a positive result indicate? It means that, this bacteria has catalase, which can also tell us which bacteria that would be. So I'm going to leave you to figure out which bacterium would have a positive result. If you're lost, look in lab 8. How do you label your plates so you can retrieve them at your next class? I feel like this is a huge gimme. This is just, aside from putting your section, what are 2 other things that you would include so that next class you can tell that that's your plate and pick it up and continue with your experiments? 14, what is the Kirby-Bauer method? I don't think we called it the Kirby-Bauer method in class, but it was when we did this. So this is, we had our plate, and remember 1 week we created this bacterial lawn using a bacterial spreader. So we covered this whole plate in bacteria, and then what I did after you left class was I put these discs down, and then the following class we could see whether or not there was inhibition. So this is the Kirby-Bauer method, okay? So this area, there is no bacteria growing because it is being inhibited by the antibiotics in this disc, okay? Whereas the bacteria is completely growing all the way up to this disc, which means that the antibiotics in this disc did not inhibit the bacteria here. So if I was looking at this plate, I could see, this is my zone of inhibition. This is my antibiotic disc, and then all of this, kind of yellow-green is our bacterial lawn, okay? Let me just make sure I answered all parts of this question. Yep. Yeah, so how can you tell if the bacteria is susceptible to the antibiotic or resistant? So, if the bacteria is growing all the way up to this disc, then it is resistant. The bacteria that is growing on this plate is resistant because it doesn't care that this disc is here. And then here, we know it's susceptible. The bacteria that's growing here is susceptible to this antibiotic because it stopped growing. It said, we're not going to get anywhere closer. We're not going to get near this thing at all, okay? So, I feel like 15 I kind of answered in that explanation. Remember that we had different letters on the top to denote something. How are fungi different from human cells? Okay, so we know that fungi are also eukaryotes. This is from Lab 5. Um, but how are they different? Do they still have mitochondria? Do they have cytoskeletons? Do they have vacuoles? Do they have endoplasmic reticulum? How are fungi different from human cells? There's one important thing that I want you to be able to express. Okay, what is candidiasis? We didn't get to talk about this. It is also from Lab 5. Okay, so if you go back to Lab 5, I believe it's the second page of that lab. Candida is a type of fungi. Okay, so it's from that eukaryotes lab. The definition of candidiasis is in that lab. What is a vacuole? A Golgi apparatus, endoplasmic reticulum, ribosome. Another thing to know is, the cytosol and the cytoskeleton. All of this is, I believe, also in Lab 5. And also, this is something that you guys should have covered in micro lecture, like first or second day of class. These are just components of the cell. What is a vacuole? What is the Golgi apparatus? So a vacuole can be, it's kind of like a bubble inside the cell, and it can have a variety of functions, but like one of the functions of one of the types of vacuoles would be a lysosome, which is filled with free radicals. And basically, what it does is anytime that something needs to be digested in the cell, like old proteins and things like that, they get directed to these lysosomes, and once they're inside, the free radicals kind of shred the proteins so that they can be recycled and everything. So just know what a vacuole is. A Golgi apparatus we kind of refer to as like the post office of the cell. That's where a lot of large molecules go to. They can be modified and then they are sent on to wherever their final destination in the cell is supposed to be. The endoplasmic reticulum is a place where proteins are made. There's a smooth ER and a rough ER. The rough ER is covered in ribosomes that make proteins. And then the ribosomes, they can be part of the rough ER. They can also be free floating in the cytosol. And so what happens when we have transcription and translation, again, something that you guys probably already covered in micro lecture, but you have your DNA in your cells, and the DNA in the nucleus is, it goes through transcription and it becomes mRNA. And then that mRNA is taken from the nucleus out into the cytosol. And then it's fed into these ribosomes. And what the ribosomes do is they basically they read the mRNA code. And while they're reading the code, they're pulling amino acids in that are attached to tRNA and build a protein. And so they end up spitting out this protein, and the protein folds, and then it is directed to wherever it needs to go in the cell. So just make sure that you know the major components of a cell. Steps of washing your hands was on exam 1. We wanted to include it on exam 2, partially because we feel like this is just a gimme, and partially because it's important that you know how to do this, especially during COVID, right? What is hemolysis? We kind of talked about this, and we talked about how they look already. So there's going to be a couple of questions on the exam that are based on hemolysis. Be able to Identify the TSI slants using the codes A, K, plus and minus. Be able to look at several slants and tell me what the code is. Okay, so we went over this yesterday, and I told you guys like the best way to study for it is to just make a chart. It's easiest to commit to memory. So remember that the first letter, this letter right here, can be A or K. A is going to be yellow and K is going to be red, or you can say, pink, but I think the exam wants you to say red. Basically, like this is asking the bacteria that are, on the TSI slant, can they ferment any of the sugars that are in the TSI? Remember, this is the triple sugar iron slant. So there's 3 different types of sugar. If any of the 3 sugars can be used by the bacteria, then you're going to have an A here. You would have a K here, and it would be red if only dextrose could be processed by the bacteria. Okay, K, this second one, it can be A or K. A is going to be a yellow butt, and K is going to be a red butt. Again, A is just going to be, in the butt of the TSI slant, can these bacteria process any of these sugars? K is going to be, there's no processing of sugars. Okay, and then this plus or minus, the first one, is does it reduce sulfur? Okay, so remember, if there's a plus sign, it means that we are observing a black precipitate in the butt. Okay, and also, if the butt is black, you can't observe whether or not the butt is red or yellow. We're going to assume that it is an acidic environment. So if you have a positive sign and there's precipitate, you know that this is also going to be an A, okay? And then does it produce gas? That's going to be this final minus sign over here. So if it produces gas, you're going to see like big fissures or cracks, or the very bottom of the agar might be lifted off of the test tube, okay? That means that there's gas being produced, which means there would be a plus sign. If it doesn't produce gas, it would be a minus sign, okay? What is an enterotube and why is it helpful? We kind of went over this. Yesterday. So an enterotube is helpful because you can run a bunch of tests at one time. So it really saves time, especially in a hospital setting, because then you don't need to have like a ton of petri dishes sitting around and stacking up and everything. You have this one enterotube with a bunch of different gels in it, and you can run a bunch of tests at one time and really figure out which kind of bacteria you're dealing with. Okay. What makes for a good antibiotic? Okay, so the answer to this is going to be on page 58 at the end of the first paragraph. Okay, we just want to know like pretty roundabout, like, does a good antibiotic kill all bacteria? Does it kill some bacteria? If you look at page 58 at the end of the first paragraph, you'll get some clarification on that. What are the definitions for prevalence and incidence? I'm not going to go over that because it's in your lab manual. You've had to look it up a couple of times. Just know what is the difference between prevalence and incidence, okay? What are the scientific names for red blood cells, white blood cells, and platelets? Remember that I told you guys in class that we don't want you to call it red blood cell or white blood cell or platelet on the exam. We want you to use the actual scientific names for these, okay? What is MRSA and what bacterium causes it? This is actually reflective of a question that is earlier that we talked about, so I'm not going to go through that. The immune response. Okay, so some classes got to go over the immune response a little bit, and some didn't. Remember that we have the adaptive and we have the innate immune response. So the innate immune response is non-specific. Okay, if some pathogen comes into your body, then your innate immune response will say, like, we don't care who you are, we're attacking you right now. So it's not specific, and therefore it's very quick. It can, it's like the first response. Okay. The adaptive immune response is a little slower because it's very specific. So remember that our body, once we're introduced to a pathogen, it can create these B cells that are specific to that pathogen, and that the B cells will secrete antibodies. Okay, so the antibodies are also very specific to whatever that pathogen is. Okay, so that's the immune response. So if you think about inflammation, would you say that inflammation is a part of the adaptive or the innate immune response? Is it specific? Is it non-specific? Can it just occur because, any pathogen comes into the body? Would you say that it is quick or slow? And I think that those clues will help you figure out whether or not inflammation is part of the innate or adaptive immune response. What is an opportunistic pathogen? So we kind of talked about this a little bit in class. Opportunistic pathogens are often just a part of our normal microflora. They don't cause any problems. The problems occur when you wipe out some of their competitors, or they're, moved to another part of the body or something. So, for example, we talked about C. diff, right? If somebody goes into the hospital and they go through a bunch of rounds of antibiotics, then those antibiotics are going to kill a lot of the bacterial species that are inside their gut. And what happens is now the ones that are resistant to those antibiotics are the only ones left, and they don't have to compete for food or anything anymore. And so what happens is they can actually kind of get out of hand and cause disease. Okay. Review identifying bacteria, looking through a microscope based on morphology. So remember, the very beginning of the semester, we were looking at bacteria, through a microscope, and sometimes we would see chains, sometimes we'd see clusters. Okay, so that's how we think of bacteria in terms of like how it will pair up with other bacteria of the same species, but also think about, like, what does each individual bacteria look like? Would you say it's bacillus? Would you say that it's, like cocci? Are they circular shaped? Okay, so definitely review the shapes of individual bacteria, and then how they form together. Are they chains? Are they clusters? Are they spirals? Like, what are they? Okay, so make sure that you review that. If you have any other questions, just feel free to reach out to us, and we will try to get your questions answered quickly. Okay, good luck.